Hello, my name is Jian, and I'm here to present to you the detection of novel microRNAs in tobacco targeting black shank and powder mildew genome. Outline of this presentation is economic impacts of fungal and oomycete diseases in tobacco, black shank and powder mildew diseases, microRNAs, experimental design and results, discussion and conclusion. First, economic impacts of fungal and oomycete diseases in tobacco. Yield loss, quality degradation, increased production cost, soil infrastructure management cost, and investment risk and uncertainty. First disease is black shank. It causes sudden wilting. The most prominent symptom is the sudden yellowing and wilting of leaves. The plant permanently wilts and dies as the infection progresses. The stem tissue near the soil line turns black. When the stalk is cut lengthwise, the pith shows decayed, dark cross sections. High soil moisture and warm weather triggers the disease. Fields with poor drainage are highly susceptible. Powder mildew. This is an airborne fungal disease. It typically starts on the lower leaves of the plant and can spread to cover the entire plant under favorable conditions. Unlike black shank, it rarely kills the plant. Here you can see white powder growth and lesion development. It damages the leaf. MicroRNAs. They are small non-coding RNAs, approximately 20 to 24 nucleotides in length, processed from a double-stranded precursor structure into a single-stranded mature microRNA. Their primary function is post-transcriptional suppression of gene expression, and a single microRNA can target multiple different mRNAs. The aim of this study is to identify novel microRNAs targeting powder mildew and black shank in Nicotiana tabacum by using in silico analysis. The experimental design for this study is first acquisition of plant microRNAs from Mirbase database and after that blast analysis, identification of mismatches and screening process, RNA fold analysis, and lastly target prediction in viral genomes. Acquisition of plant microRNAs from Mirbase database. Major microRNA sequences of all registered plant species were retrieved from the Mirbase database. To prevent repetitive analysis and false positives, duplicate or highly similar sequences were filtered out. Excel conditional formatting was used to remove redundancy. A non-redundant dataset, aka singletons, was established for further analysis. Mismatches and blast analysis. On the right, you can see my BLASTAM parameters. I used NCBI BLASTAM program, and my target organism was Nicotiana tabacum. Candidate sequences were manually aligned with the reference microRNA sequences. A maximum of 7 mismatches and/or indels were allowed between the reference and the target ESD sequences. RNA fold analysis. I used RNA fold web server VNR RNA package for my secondary structure drawings. And there is 4 criteria for this secondary structure. First one is the potential microRNA sequence must be located at a suitable position within the stem loop hairpin structure. The second criteria is the mature microRNA and microRNA sequences must not contain any loops or breaks within their paired regions. Third one is thermodynamic thresholds. Minimum free energy must be less than or equal minus 18 kcal mole. Minimum falling energy index value must be bigger than 0.85. Fourth criteria is pairing requirements. A minimum of 12 Watson Crick base pairs must exist between the mature microRNA and the microRNA sequences. Results: the microRNAs retrieved from the Mirbase database were reduced to 6083 mature microRNAs after the singleton processing. Sequences compared against tobacco genes via NCBI BLASTM. 280 mRNAs met the criteria. Here you can see secondary structure results for black shank. There is 75 secondary structure for black shank. RNA hybrid results for black shank. And secondary structure results for powdery mildew. There are 15 secondary structures that met the criteria for powdery mildew. And RNA hybrid results for powdery mildew. Discussion. There's two articles in discussion. First one is for black shank and tobacco. This study aimed to uncover the role of microRNAs in tobacco's response to black shank disease and to provide new genetic resources for breeding-resistant tobacco. They identified a specific microRNA whose production was significantly induced in tobacco plants infected with the pathogen. They created a tobacco plants that constantly produced this microRNA at high levels and challenged them with the pathogen. They examined the effects of this microRNA overexpression on the plant's overall development. The study demonstrated that this microRNA plays a critical role in increasing resistance to black shank in tobacco plants, but this comes with a cost to plant development. The second article is about powdery mildew infection in cucumber. This study aimed to understand the role played by microRNAs in defense mechanisms against powdery mildew disease in cucumber plants and to comprehensively map the gene regulatory network that responds to this pathogen. The study used the small RNA sequencing technique to identify all small RNAs, including microRNAs, in cucumber leaves after infection. Degradome sequencing was used to confirm which genes the microRNAs cleave and silence. This method allows for the precise determination of the cleavage points of microRNA target genes. The biological functions of the genes targeted by the microRNAs were analyzed. The studies successfully mapped the comprehensive microRNA mediated gene regulatory network in cucumber in response to powdery mildew, highlighting critical defense and signaling pathways for developing disease resistance varieties. 118 black shank and 25 powdery mildew microRNAs are predicted to target the tobacco genome. High number of stable secondary structures supports the functional activity and stability of these targeting microRNAs. This work provides in silico evidence supporting the cross-kingdom RNA interference mechanism in tobacco-pathogen interactions. These results open doors for developing microRNA-based genetic resistance strategies against both diseases. Thank you for listening.